There's a cat living in the rainforests of Central Africa that most people have never heard of. It is not a lion. It is not a leopard. It is a medium-sized, reclusive predator called the African golden cat. And for most of its existence, it has moved through dense forest canopy so quietly that field researchers sometimes go entire careers without seeing one in the wild. Scientists studying it did not expect to find anything connecting it to the animal sleeping on your sofa. But when a team of geneticists ran a sweeping comparison of black coat DNA across every wild cat species, on Earth, the African golden cat came back with a result that stopped the analysis cold. Out of 37 wildcat species, out of 13 separate evolutionary events that independently produced black coloration across the entire cat family, only one wild species carried a melanistic mutation that closely matched the domestic black cat. It was not the leopard. It was not the jaguar. It was a forest cat most people cannot name. That result raised questions that went far beyond coat color. To understand why the match matters, you first have to understand how unusual black coloration actually is in wildcats. Walk into any animal shelter and black cats are everywhere, so common they barely attract attention. But in nature, solid black coloration across the entire coat is extraordinarily rare among wild felids. The 37 wildcat species alive today are overwhelmingly patterned animals: spots, rosettes, stripes, tawny gradients, countershading. These are the standard visual signatures of wild cats across every continent. The patterns serve functional purposes, breaking up the animal's outline in its environment and allowing it to approach prey or avoid larger predators without being seen. Solid black coloration works differently. It does not break up an outline. It erases it entirely. And whether that is an advantage or a liability depends entirely on the environment. The genetic machinery that produces black coloration in cats runs through two primary signaling genes. One gene functions as an accelerator, pushing pigment-producing cells toward dark pigment output. The other functions as a suppressor, holding that drive in check and allowing lighter pigments to emerge. Normally, these two systems balance against each other, producing the range of tawny, spotted, and striped coats typical of wildcats. Black coloration appears when the suppressor gene breaks down. With the break-off, the accelerator runs continuously, flooding hair follicles with dark pigment and producing a coat that reads as solid black from root to tip. The specific mutations that disable the suppressor gene are the genetic signatures researchers track when they map melanism across species. What the mapping revealed was not what most researchers had assumed going in. The expectation was that black coloration in wildcats had probably evolved a handful of times and spread through the family via shared ancestry. What the data showed instead was 13 independent origins, 13 separate mutations in 13 different branches of the cat family tree, each disabling the pigment suppressor through a slightly different molecular change, each producing the same visible outcome through a distinct genetic route. According to research published in comparative felid genomics literature, this degree of convergent evolution in a single visible trait is unusual even among mammals, which frequently rediscover successful adaptations across unrelated lineages. The cat family, it turned out, had been solving the same problem over and over for millions of years, arriving at the same answer each time through whatever genetic pathway happened to be available in that particular species. The pattern told researchers something important before they had even compared a single mutation to the domestic cat. When a trait evolves 13 times independently, natural selection is not operating randomly. Something about being black was worth finding repeatedly across different continents, different environments, and different branches of a family tree that diverged millions of years ago. The question was what that advantage was, and whether the domestic black cat's version of the mutation was connected to any of the wild versions in a meaningful way. Researchers from institutions including those contributing to the feline genome project began assembling a catalog. Every wild cat species known to produce melanistic individuals was included. The leopard, whose black variant is famous enough to have its own common name. The jaguar, whose black individuals roam the Amazon basin in higher proportions than most people realize. The serval, a long-legged African cat that occasionally produces dark individuals in highland forest zones. Several South American small cat species, including the oncilla, whose melanistic form appears in certain forest populations at measurable frequency, and dozens of others, each with its melanistic mutation characterized, sequenced, and placed into the comparison. The domestic black cat's mutation was already well characterized from prior work. The team placed it at the center of the comparison and began measuring genetic distance. Most of the wildcat mutations came back clearly distinct, different positions in the suppressor gene, different mechanisms of disruption, different downstream effects on how the gene's protein product behaved. The catalog was comprehensive and the differences were real. 13 evolutionary events had produced 13 variants, and most of them, when held against the domestic cat's version, looked like distant cousins at best. The African golden cat is not an animal that appears in wildlife documentaries with any regularity. It does not have the cultural presence of the leopard or the dramatic mythology of the black panther. It lives in the equatorial rainforest belt, stretching across Central and West Africa, from Senegal in the west to Kenya in the east, moving through dense undergrowth at night and disappearing into canopy shadow during the day. Camera trap studies conducted by researchers at institutions, including the Wildlife Conservation Society, have described it as one of the least photographed wildcat species on Earth. Relative to its range size, its coat in typical individuals runs from golden brown to slate gray, sometimes reddish, sometimes almost charcoal without being fully melanistic. But in certain forest populations, particularly in the dense interior zones of the Congo Basin, black individuals appear at frequencies that astonished the researchers who first documented them systematically. Some survey sites recorded melanistic African golden cats at rates exceeding 50% of all individuals detected. In a wildcat species, that is an extraordinary proportion. For context, melanistic leopards in most African populations appear at rates well below 10%. The African golden cat was producing black individuals at roughly 5 times that rate in its core forest habitat, which suggested that whatever advantage black coloration provided in that environment, this species was experiencing it more intensely than almost any other wild felid on Earth. When the genetic comparison placed the African golden cat's melanistic mutation alongside the domestic black cat's mutation, the similarity was not subtle. The mutations sat at closely related positions within the suppressor gene. The molecular mechanism through which each mutation disrupted normal gene function operated through a comparable pathway. The resulting protein product in both species was altered in ways that were structurally similar. According to the comparative analysis, the genetic distance between the domestic cat's melanistic variant and the African golden cat's melanistic variant was measurably smaller than the distance between the domestic cat and any other wildcat species tested. 12 of the 13 independent evolutionary events that had produced black coloration across the cat family came back as recognizably distinct from the domestic mutation. The 13th, belonging to the African golden cat, did not. That result immediately raised the question that any rigorous comparative genetics study has to confront. Does similarity mean shared ancestry, or does it mean convergence? Two species can carry nearly identical mutations through two completely different histories. One possibility is that both lineages inherited the mutation from a common ancestor that lived before their evolutionary paths diverged. The domestic cat descends from the Near Eastern wildcat, a species whose ancestral range covered the Middle East and northeastern Africa, geographically adjacent to the range of the African golden cat's own ancestors. If the shared mutation predates the divergence of those lineages, it would represent an ancient piece of genetic code that both species inherited and maintained because it carried consistent advantages in certain environments. The other possibility is that the similarity is convergent, meaning both species independently arrived at the same molecular solution because that particular position in the suppressor gene is simply the most genetically accessible route to black coloration. The same change, appearing twice, not because it was inherited, but because the gene structure made it the path of least resistance for natural selection to exploit. Distinguishing between those two explanations requires additional phylogenetic analysis that researchers are still working through. But either answer carries significant implications. If the mutation is ancient and shared, it means black coloration in domestic cats is not a product of domestication at all. It would mean that the domestic cat's ancestors carried the genetic potential for melanism long before humans were involved, and that the same ancient variant persisted in the African golden cat lineage because forest environments kept selecting for it. If the mutation is convergent, it means the suppressor gene has a specific vulnerability at that position, a molecular weak point that natural selection keeps finding, because it produces a reliable functional outcome. That would make the African golden cat and the domestic black cat independent experiments that arrived at the same result, which is remarkable in its own right. What made the comparison more complex was the geographic logic underlying it. Melanism in wild cats does not distribute randomly across species ranges. Researchers analyzing field data from melanistic felid populations across Asia, Africa, and the Americas found a consistent environmental pattern. Black variants concentrate in humid, densely forested habitat. A study drawing on leopard population. Data across sub-Saharan Africa and Southeast Asia found that melanistic individuals were significantly more common in rainforest zones than in savanna or semi-arid habitat. The same pattern appeared in jaguar data from South America, where black individuals are documented far more frequently in the dense interior of the Amazon basin than along its drier forest margins. The serval's melanistic populations cluster in highland forest zones of East Africa, rather than in the open grasslands where the species is most commonly associated in popular imagination. The African golden cat's exceptional rate of melanism fits this pattern more completely than any other species. It is among the most forest-dependent wildcats on Earth. It does not range into open habitat the way leopards or servals do. Its entire ecological existence is structured around dense canopy cover, which means the selective pressure favoring black, coloration over spotted, or tawny coloration, has been operating on it more consistently and with fewer interruptions than in any other felid studied. Domestic cats are not forest animals. They have not been forest animals for thousands of years. They live in human homes, in urban alleyways, in agricultural landscapes, in desert villages, and Arctic research stations. The environments they occupy span nearly every climate and habitat type on Earth. And almost none of those environments resemble the dense equatorial rainforest where black coloration repeatedly proved its worth in wild felids. By the ecological logic that explains melanism in leopards and jaguars and African golden cats, black domestic cats should be rare. The selection pressure that drove black coloration to high frequency in forest wild cats simply does not exist in a kitchen or an apartment or a barn. And yet, black is one of the most common coat colors in the entire domestic cat population globally. The question of why that is turns out to have a cleaner answer than the question of how the mutation got there in the first place. Domestication. Remove the filter. In wildcat populations, living in open or semi-open environments, black coloration carries measurable costs. A solid black animal is more visible against dry grass, pale rock, and open terrain than a spotted or tawny one. Predation pressure and hunting efficiency both work against black coloration in non-forest environments, which is why melanistic individuals in open country wildcat populations remain rare even when the genetic variant is present in the gene pool. The mutation persists at low frequency because carriers occasionally survive and reproduce, but it never rises to dominance because the environment keeps selecting against it. Domestic cats, removed from that selection pressure by living alongside humans, experience no equivalent penalty for being black. A black cat in a Roman granary or an Egyptian household faced no predator that would preferentially target it over a tabby. The mutation that natural selection would have suppressed in an open country wild population was free to drift upward in frequency simply because nothing was eliminating it. Over thousands of years of domestication across dozens of human cultures, black accumulated in the domestic cat population by default. Not by design, and not by the same functional logic that drove it to high frequency in rainforest felids. That explanation accounts for the color's prevalence. It does not fully account for why the domestic mutation most closely resembles the African golden cats, rather than any of the other 12 independent melanistic variants cataloged across the cat family. And it is here that a second layer of the research opened up, one that moved the conversation away from pigmentation entirely and into immunology. Infrastructure. The same signaling cascade that determines whether a hair follicle produces dark or light pigment also influences how certain immune cells respond to pathogens. This overlap is not coincidental. It reflects the deep evolutionary history of the pathway itself, which predates the specialization of either pigment cells or immune cells in vertebrate biology. According to research published through institutions contributing to mammalian immunogenetics literature, mutations in the suppressor gene do not simply change coat color in isolation. They alter the behavior of the entire signaling pathway in every tissue where it operates. For domestic black cats, this has produced a measurable and much discussed biological footnote. Several studies examining feline disease resistance found that black domestic cats showed statistically elevated resistance to feline immunodeficiency virus compared to cats carrying other coat color genotypes. Feline immunodeficiency virus is a lentivirus closely related in structure and mechanism to HIV in humans. The resistance observed in black cats is not absolute, and researchers have been careful to note that the evidence does not support any clinical claim about black cats being immune to the disease. But the genetic mechanism underlying the association is real and traces directly to the shared molecular pathway connecting the suppressor gene to immune cell function. The mutation that disables the pigment suppressor also shifts the behavior of the immune signaling cascade in ways that appear to confer partial resistance to certain viral pathogens. Color and immunity encoded in the same gene, altered by the same mutation. The African golden cat presents a parallel finding that researchers consider significant precisely because it was not predicted before the genetic comparison was made. Field surveys and studies of captive African golden cat populations have documented that melanistic individuals appear to show lower rates of certain parasitic infections than golden-coated individuals from the same populations. The sample sizes available from a species this rarely studied are small, and the researchers involved have been explicit about treating the data as preliminary. But the directional finding aligns exactly with what the shared gene pathway would predict. If the domestic black cat and the melanistic African golden cat carry mutations at closely related positions in the same suppressor gene, and if that gene influences immune function as well as pigmentation, then the immune effects of those mutations should run in similar directions in both species. The color comparison had started as a question about fur. The immune pathway analysis turned it into a question about disease, ancient selection pressure, and whether the mutation's persistence across both lineages had ever been. There is a statistic that sits uncomfortably alongside everything the genetic comparison revealed. Black cats are adopted from animal shelters at lower rates than cats of any other color. The phenomenon is documented consistently enough across shelter population studies in multiple countries that researchers who study companion animal demographics have given it a name: black cat bias. The explanations offered are practical and mundane. Black cats are harder to photograph against dark shelter backgrounds. Their facial expressions are less readable in low light. Their appearance, in small enclosures, strikes prospective adopters as less distinctive than a tabby's stripes or a calico's patchwork. Cultural associations with black cats vary across communities, and in some contexts carry negative connotations that influence adoption decisions in ways people do not always consciously recognize. The result is that black cats spend longer in shelters than their littermates. They are the last to leave, and in overcrowded facilities operating at capacity, statistically the most likely to be euthanized when space runs out. What the genome comparison adds to that shelter statistic is not a simple rebuttal. It is a context that makes the statistic stranger than it already was. The domestic black cat carries a mutation in a suppressor gene that influences both pigmentation and immune system signaling. That mutation most closely resembles, out of every wild cat species tested, the variant found in the African golden. Cat, a species whose forest populations produce black individuals at rates exceeding 50%, because the combined advantages of camouflage and immune function have been selecting for that mutation. Continuously for longer than most other felid lineages have been distinct species. The domestic black cat's version of that mutation appears, based on current evidence, to confer measurable resistance to at least one major viral pathogen. The color that shelter adoption data consistently identifies as the least preferred is the color with the deepest functional history and the strongest candidate for carrying a genuine biological advantage within the domestic cat population. The irony is not lost on the researchers who study both sides of this picture. The most biologically resilient color variant in the global domestic cat population is the one that spends the longest time in the environment most likely to compromise biological resilience. Extended shelter stays expose cats to chronic psychological stress, elevated cortisol levels, disrupted sleep cycles, and is notable when it appears twice or 3 times in distantly related lineages, 13 times within a single family, across millions of years on multiple continents, through multiple distinct genetic mechanisms, suggests that the selective pressure favoring black coloration in certain environments has been operating with a consistency and force that most models do not fully account for. According to researchers contributing to feline evolutionary biology literature, the cat family may represent one of the clearest documented cases of repeated phenotypic convergence, driven by an identifiable and consistent ecological gradient, the transition from open habitat to closed forest canopy. The domestic cat sits outside that gradient entirely, which makes it a unique data point in the comparison. It is the only population in which black coloration rose to high frequency in the complete absence of the selective pressure that drove it everywhere else. That absence tells researchers something about how powerfully predation pressure and habitat structure normally constrain coat color. Variation in wild felids. Remove those constraints and black accumulates rapidly, suggesting the mutation itself carries no significant biological cost in environments where visibility to predators is not a survival factor. The domestic cat became, unintentionally, a long-running natural experiment in what happens to a melanistic mutation when natural selection stops caring about it. What remains open is the origin question: whether the domestic black cat and the African golden cat share their mutation through ancient common ancestry or arrived at it independently through convergent. Pressure on the same molecular target is a question that current phylogenetic data has not fully resolved. Researchers working on expanded felid genome sequencing projects are building the datasets that will eventually answer it. The answer will matter, not because it changes anything about the cats themselves, but because it will clarify whether the immune pathway advantages associated with the mutation have a single deep origin, or whether natural selection found the same functional solution twice through entirely separate routes. Either answer is remarkable. A mutation that evolved once and persisted across tens of millions of years in 2 wildly different species because it kept proving useful would be remarkable. A mutation that evolved twice at the same position in the same gene, because that position is simply the most accessible genetic route to a suite of connected advantages, would be equally remarkable. The domestics, black cat and the African golden cat, are not closely related. They do not share a habitat. They do not share a history that any living observer would recognize. What they share is a change in a single gene, sitting at a position where that change keeps mattering, across environments and centuries in every category of difference that separates a rainforest predator from an animal sleeping on a windowsill. Science found the connection. The question of what to do with it belongs to everyone watching.